Good afternoon, folks. Welcome back.
Hello.
Hello.
So thanks for coming out here this afternoon. And we'll get started in just a second here as I get everybody logged in. Okay, so welcome back, folks. And good afternoon to you. I hope that you're well on your way to organizing your Thanksgiving plans and you get a little bit of downtime. Okay, so let's continue here. We are going to finish off our coverage of Chapter 6 today. And so that means this coming Tuesday, I'll be starting Chapter 14. Now, Chapter 14, as you know, is not going to be part of Lecture Exam 3. So when we cover Chapter 6 and finish it today, we have finished the content for Lecture Exam 3. So hold on just a second, folks. I have a couple messages that I need to read in the chat window. Okay. Tiffany, I have your message. I understand. Thank you. Yasmin, yes, Yasmin, I have received your email. Thank you for that. Okay. All right. So yes, we're going to finish Chapter 6 next Tuesday, start Chapter 14. Chapter 14 will be the first chapter that will be covered on your final exam. Speaking of the final exam, I finally have the final exam information. So I'll keep reminding you about this, but let me put the final exam date and time into the chat window for you, okay? All right, so the final exam for this class is Thursday, December 16th. And it's at normal class time, 12:30. Okay, final exam is Thursday, December 16th at 12:30. And I will, I will let you know that you will have a 2-hour block of time for the exam. Final exam timing is set by the institution. And so HAC wants all students to have 2 hours to take their final exams, okay? So the final exam, Thursday, December 16th, starting at 12:30, you have 2 hours for that, okay? All right, do you have any questions before we continue? Okay, so Lena is asking if the final exam is cumulative. No, it is not. The final exam is not cumulative. The final exam is just the final 3 chapters of the semester. Okay, that would be chapters 14, 15, and 17. Okay, any other questions? Okay, gang. All right, so tell you what, please get out your workbook assignment number 4. We're going to go through that and see how you made out with that. I have a feeling that many of you did just quite okay with it, which is good. So get out your workbook assignment number 4, and let me bring it up on my screen. There we go. Okay.
Professor, I actually did have a quick question about the test. I just thought of it. For our final, are we going to have essays as well? So it's just going to be like a regular test of yours?
Yes, it will be a typical normal exam that you've taken all semester long. There's nothing special about it just because we're calling it the final exam. It is not cumulative. It will be a standard traditional lecture exam covering the content of chapters 14, 15, and 17. It will have multiple choice. It will have fill in the blank. It will have essays, yes.
Okay, thank you so much.
Okay.
Is that going to be on class marker as well?
It will be on class marker, that's correct. Okay, anything else? Okay. So since we're talking about these topics, it reminds me that later this afternoon, I will email to you, in addition to the normal class notes today, I will email you the study guide for lecture exam 3. So that'll be coming out this afternoon, okay? All right, lots of good things on our schedule here. All right, so this is the workbook assignment that I'd asked you to wrestle with for a few days. Okay, so let me just review what's being asked, and then I'll ask for you to contribute in the chat window. So the scenario is that you're working for a biotech company that manufactures insulin and sells that insulin to pharmacies. Your supervisor tells you to begin a culture of E. coli the night before, so that the next day you can purify it. Okay. You start growing the culture at 5 o'clock in the evening. You seed the initial culture with 250,000 cells that have just entered log phase. Now, I realize we haven't really talked about log phase yet. That's on my agenda today. At 7 o'clock that same evening, you're home playing a video game. You remember that this strain of E. coli will only stay in log phase for 12 generations. And you know that because you did previous studies with this bacterium. You know from previous work that the generation time is 27.8 minutes per generation. And you've got to start purifying insulin before the cells leave log phase, because when you leave log phase, the cells get a little bit sicker. And when they get sicker, the amount of product you can purify from them is not as much. Okay, and we'll talk about that in a little bit. So, okay. So the first question here, what time will it be when these cells transition from log to stationary phase growth? Go ahead and put your response in to everybody. Let's see what everybody is saying about this. Okay. All right. So go ahead and type your responses in there. All right. And go ahead and start scanning the responses up and down. Okay, up and down. And so, you know, as I look at it, I'm seeing a lot of consistency, but not quite 100% consistency. Okay, not quite 100% consistency. So, all right, so I suppose we need to talk about that, right? The answer would be, and I'm just rounding it here, the answer would be roughly 10:30, but if you did all the unit conversion stuff, that would probably extend out to about 10:33 in the evening. Okay, about 10:33 in the evening. So does anybody need the logic of that covered? Or let me put it this way, would one of you be willing to explain in just simple language, don't have to get very technical, just explain the reasoning process you went through to get to your answer?
I will.
Okay, thank you. Go ahead.
So it took 27.8 minutes to generate one, or per generation. So if the log phase went through 12 generations, I multiplied 27.8 minutes by 12 generations and got 333.6 minutes. And then I divided that by 60 minutes per hour and got 5.56 hours. And then had to kind of figure out what the 0.56 hours would be in minutes. So 0.5 is about 30 minutes. And then I just kind of, you know, rounded around to get to 10:33 p.m.
Okay, thank you, Rachel. That's fine. And so I think the sticking point for some students once in a while is if you have, you know, one of your computations with there, when you took the total minutes of growth, 333.6 minutes, and you divided by 60, you got an answer of 5.56 hours. 0.56 is roughly a half an hour, right? It is not 56 minutes. So you have to be very careful here when you're doing your calculations that you don't think 5.56 hours is 5 hours and 56 minutes. It's not 5 hours and 56 minutes. It's 5 hours and 56 hundredths of an hour, right? So 5.56 is roughly 5.5 hours. Okay. Anybody out there in the cyberverse would like to ask a question about that, or would like me to go through the process? If you want me to write it down on the screen, I'll do that if you want me to do that. Okay, all right. If you're sitting at home and you're thinking, you know, I'm just too embarrassed to say that I'd want help on this, please come see me during office hours. Okay, I do want you to understand this. All right, so absolutely. So the time will be roughly 10:33 in the evening, and that is the correct answer. All right, question 2, as protein purification is a multi-step, multi-hour process, do you think you will get a full night's sleep before having to start your shift the next day? And so obviously there's no real math involved here, but thanks for those contributing a response in the chat window, right? I appreciate that probably very few of you, if any of you, have actually ever purified a protein in a lab. Protein purification can be very arduous work, very time consuming, and it's something you can't rush because, you know, it's kind of like when you're cooking your Thanksgiving meal. I mean, do you want to eat a turkey that was nuked for 30 minutes in the microwave, or do you want to eat from a turkey that's been basted and gently cooked in the oven for several hours, right? You get a better tasting turkey, you get a better result when you take your time at cooking rather than shoving it in the microwave. And that's the point I'm making here. Protein purification, as an example of many laboratory procedures, is best done when you take your time at it. So, so if you kind of made a mistake here and had to go in late at night to start the protein purification because you kind of started your cells at the wrong point, that means you're going to have a choice. You're either going to have to stay at work for a good bit into the wee hours of the morning and actually do the work, or you're going to just have to say, okay, I'm going to trash this run and I'll get in the next day and I'll just have to tell my boss that, well, I made a mistake. Okay, so that's basically the choice you have here. Okay. All right. Let's continue. Question 3. How many cells will be present at the transition point from log to stationary phase growth? What did you come up with, gang? Okay. All right. Things are looking pretty good here, gang. Excellent. Awesome. I'm not seeing anything that's alarming to me. Okay. So I think all of you have typed in the correct answer. Very good. Yeah. 1,024,000,000. 1,024,000,000 cells is the correct answer for question 3. Good job. All right. Any questions about that? All right, let's see how you made out with question 4. Okay, question 4. If each E. coli cell contains 4.8 micrograms of insulin, how many grams of insulin will you recover from all the cells present at the log to stationary phase transition point? Okay, so just go ahead and take a look at your classmates' answers, see if they jive with your answers. Okay. All right, so just about everybody is in agreement here. The correct answer is 4,915.2 grams. Okay, 4,915.2 grams is the correct answer. So
Could we
Arlen,
go over that one, please?
yes, we will. Arlen, can you please speak to this, how you got your answer?
Actually, Rachel helped me come to this. She would be able to probably explain it better because I got confused because I forgot to actually do it at first to convert it.
Okay, So Rachel's already contributed, and I appreciate that. Is another student that would be willing to walk through this? Come on, folks.
I
Yeah,
can try.
I can.
Oh, well, go ahead. No,
Okay. So you take your answer from number 3 and just multiply that by 4.8. And then you get the amount of micrograms of insulin that all those cells contain. And then you have to convert that micrograms into grams, which is 1,000,000. So you just divide that answer by 1,000,000 because there's 1,000,000 micrograms in a gram. You get
Okay.
the right answer.
Okay, absolutely. Just because there was a,
Sorry.
go ahead, Elena.
I actually worked with a tutor on this, and I don't know, we just set it up differently, and that's why I got my answer. I didn't get the 0.2. So what I did is, so the way we worked it is 4.8 micrograms times 1 gram over 1,000 micrograms. We eliminated the micrograms and we got 0.0048 and then multiplied that by the number of cells.
Okay,
So where did we go wrong there?
I'm
You
not
don't want to
sure.
multiply. You want to divide.
So
the
Elena, let me
I'm going
walk
to
it through here.
microgram.
Hold on, folks, hold on. Let me walk it through here, okay? All right, so as Taylor was indicating, right, you take your answer for number 3, which is 1,024,000,000 cells, and you multiply it by 4.8 micrograms of insulin per cell. Clearly the cells cross out, and that gives you roughly, and I'll just round it here, 4.9 times 10 to the 9th micrograms, okay? And so at this point, it's just a unit conversion, correct? So we need to go from 4.9 micrograms, 4.9 times 10 to the 9th micrograms to grams, right? So 4.9 times 10 to the 9th. Okay, so micrograms over 1 gram, and we have 1,000,000 here, 1 times 10 to the 6th, right? So that will give you 4,915.2 grams. Okay, all right. Elena, is this logic working for you?
Yeah, I understand that. I just don't understand why me and the tutor came up with something else. I mean, I
So-
got more or less the kind of the same numbers, but it was just, different. It was a decimal point and everything. We just set it up differently. And that
Okay.
makes the answer wrong. So then that makes my next answer wrong as well.
Right, absolutely. So Elena, what I'd like to do with you is if I could get together with you after lab for like 2 minutes and be able to, if I can see your work or if we can reproduce it on the screen, then we can work through that, okay?
Okay, that's fine.
All right,
You want to go in between lab or after lab?
after lab, please.
Okay, got it.
All right.
Thank you.
Okay, so there we have our answer for that. Okay, and question 5. What'd you get for question 5, gang? If you sell the insulin to pharmacies at a price of $5 per gram, how much money would you make if you sold all the insulin, right? So you take your answer from question 4, you multiply it by $5 per gram, and you get $24,576. Not a bad profit there, not a bad revenue stream, okay? You're absolutely right. 24,576 is the correct answer. And so, most of you are never going to be in this position, right? Because you're going off to do things in medical careers, okay? But somebody that's going into the scientific arena, once again, if you start your experiment wrong, as is suggested with the earlier questions, right? Question number 2 asked you, do you think you will get a full night's sleep? The issue is, if you don't set these experiments up right during a production run, you're going to be working in the middle of the night, or, as I said, you're going to have to tell your boss the next day, I screwed up and I didn't set the experiment up properly. And what I'm saying to you is that if you take the second option and you say, well, I'm going to go home and sleep because I'm tired, I'll just tell my boss I made a mistake. What are you basically admitting to? You're basically admitting that you've just cost the company a production run of $24,000. Boss is not going to be too happy about that, right? Because that's how the real world actually works. Okay. So, okay. Folks, I have pretty good feeling about this. It seems that most of you went through this just fine. So, I'm going to help Elena. Are there any other questions that you might have about this?
I'm okay. I just, you know how I am with this math conversion. We've been here before.
Yep, absolutely, Elena, and I'm definitely going to help you. I just don't want to take all of the class time for that, but I will spend time with you.
Oh yeah, I got it.
Okay. All right, folks. So good job on that, and expect that in this afternoon's email, not only will you have class notes, you will have the study guide. There'll be another math problem coming your way. Okay, so I'm going to keep hitting you with math problems up until the point of the actual exam, so you get that practice and build a level of confidence in yourself. Okay. All right. Let's continue. Let's continue. Okay, so last time we were in chapter 6 and we were talking about the physical requirements for growth. Okay, so I'm going to move beyond that section of text and start a new section on the chemical requirements for growth. Okay, the chemical requirements for growth. All right, so it is somewhat unfortunate that we're not actually physically in a lab together this semester, because this chapter does present some of the ideas that would have come about by doing actual lab work at school, and you're just not getting that experience this year or this semester, okay? But we'll do the best we can here. When we talk about the chemical requirements for growth, what we're going to do is, identify some of the major chemical elements that are the building blocks for obviously important molecules that allow life to be possible on planet Earth. Probably and arguably the biggest one is carbon, right? There's an entire field of chemistry called organic chemistry, which someday some of you folks, for better or worse, might be required to take. And it is important. It's a challenging topic, but it is important because all of the major categories of organic macromolecules you learned about in chapter 2 of this course, carbohydrates, proteins, lipids, nucleic acids, right? They are all based on molecules where the skeleton of the molecule is a chain of linked carbon atoms, okay? So carbon is critical to build all 4 major categories of organic macromolecules, okay? Nitrogen. Well, nitrogen, I'm not talking about nitrogen gas, I'm talking about the elements here. This is what we're talking about, the elements, okay? So this is an element discussion. Nitrogen as an element is incorporated and used to build very important things like nucleotides, right? If you remember us talking a little bit about the chemical makeup of nucleotides and the chemical makeup of amino acids, you will find nitrogen atoms present. Okay, so the chemistry of life on this planet would not be possible without nitrogen atoms. Sulfur. Sulfur is used in a variety of important molecules. You're going to find sulfur in some amino acids. And interestingly enough, you will also find sulfur in some of the vitamins that act as coenzymes to drive the metabolism of the cell forward. Okay. So sulfur is also very important. Phosphorus. Okay, phosphorus is another element. Phosphorus, as you can probably guess at this point, is absolutely critical for things like lipids, right? Think about phospholipids that are used to build the plasma membrane of every kind of cell on the planet. So this is needed for phospholipids. It's also going to be needed for molecules like ATP, right? Adenosine triphosphate. And you're also going to need phosphorus to manufacture your nucleotides, DNA and RNA nucleotides. Okay. None of these things should really be a mystery to you. They would only be a mystery if you've never looked at the chemical structure of some of these categories of molecules that we're talking about. And at least to a very first basic approximation, I know that you did see some of these structures when you looked at Chapter 2 this semester. Okay, well, oxygen. Oxygen is usually important, right? I mean, now we think about the oxygen we breathe in the atmosphere, that's diatomic oxygen, it's O2, okay? But obviously individual oxygen atoms are needed. The environment that we live in, our home, our planet Earth, has to provide oxygen atoms that can be linked to build oxygen gas that allows us to breathe. And you know from your time studying Chapter 5 metabolism that oxygen O2 is the terminal electron acceptor at the end of the electron transport chain. And without that, we wouldn't have a way to efficiently manufacture ATP. Okay, so this is needed to breathe and critical to the efficiency of the electron transport chain. Okay. There are other trace elements that I'm not going to go into great detail about trace elements, but when I say trace elements, I'm talking about some of the things that you would find in a vitamin supplement at your supermarket, things like iron, copper, zinc, molybdenum, things like this. These are elements on the periodic table. These are cofactors that help support the behavior and activity of the holoenzymes that drive metabolism forward. All right, so you know, we're kind of stating the obvious here. We're preaching to the choir that these elements are absolutely critical for life on our planet, whether it be microbial life of bacteria, protozoa, fungi, or higher eukaryotic life such as animal life, us as humans, the animals, plants, things like that. Absolutely critical for all of that. Okay. So hopefully that's not something that bothers you too much. Please ask questions if you have it. And so what we're going to do is transition the conversation into some of the other talking points in this chapter. So I'm at page 159 in the textbook, somewhere around table 6.2. And it's in this chapter that the textbook presents to you different kinds of media. And again, it's very unfortunate because you would have been able to use some of this media in lab this semester if we would physically be present in lab. You're not going to get that experience, unfortunately, this semester. but let's talk about different types of culture media. Different types of culture media. And so when I say media, when the book says media, it's basically a scientific way of talking about food. We're talking about the kind of food that bacteria and other types of microorganisms like to consume to support their metabolism so they can live and eventually grow and reproduce. So media is basically food, okay? All right, so there's several different kinds of media you would use in a typical microbiology lab. First, we have chemically defined media. Okay, so table 6.2 shows you an example of a chemically defined media recipe that could be used to grow something like E. coli. I mean, don't bother trying to memorize these recipes. That's why we have microbiology recipe cookbooks to make up these medias, and you bring it up on your computer or you pull the book off of the shelf and you make these things up to grow the microorganisms. But just take a look at some of the material in table 6.2. You would need glucose, ammonium phosphate, sodium chloride, magnesium sulfate, potassium phosphate, and of course, all of that would be dissolved in water. Those are the nutrients that E. coli would need to live off of this food supply. If you take a look at table 6.3, here's a different kind of chemically defined media for a different microorganism. I'm not even Leuconostoc. I won't read that recipe, but just glance over table 6.3 and you can see a variety of salts, amino acids, various nitrogen bases, right? The adenine, guanine, uracil, and xanthine, vitamins, trace elements, sodium acetate. So this is the identity and quantity of different molecules dissolved in water. Victoria, I see your comment. It's okay. Just do the best you can. All right. So chemically defined media is media, meaning food.
Dr. Allen, your screen is lagging.
It's lagging. Okay.
Yeah, like I can hear you typing. You yourself are fine, but your screen share is lagging.
Okay, let me just ask in the chat window. Is everyone noticing a lag in my screen share? Okay. All right, folks. Tell you what I'm going to do. I hate to do it, but for the moment, I hate to do it, but for the moment, I'll turn my video off. Hopefully that'll improve my internet bandwidth enough so that the note taking is less choppy. So let's try that for a little bit, okay? Hold on a sec. Okay, so let's try this for a little bit. And so, can you hear me okay right now? Am I still coming through? Can you hear the audio?
Yes.
Okay, thank you. Okay, so let's continue, folks. So chemically defined media, and I'll reference tables 6.2 and 6.3. This is media in which the identity and concentration of every chemical present in the food is known. Okay, and so this type of media, This type of media tends to be very expensive because, you know, you have to know exactly what's in the media. And that makes preparation of the media tedious, laborious, and therefore expensive to do, expensive to manufacture. This is expensive media, okay? It also happens to be the media that that provides the best reproducibility of experiments in a scientific setting. Okay. All right, so stay with me here, folks, if you can. I apologize for the lag. I don't know what to say at this time. Hopefully it will get better. So let's continue here. I'm driving towards a point with my conversation here. I totally get that none of you have experience with these food sources that I'm talking about. So that makes it a little bit more nebulous to try to make the point that I want to make. But we'll start with number one is the chemically defined media. In this kind of food, you know exactly what's in it. And because it's laborious to prepare this kind of food, so you know exactly what's in it, it's very expensive to make. Even though it's expensive, because you know what's in it, it provides the best environment for scientific reproducibility. Okay. Now, an alternative food source would be a complex media. Okay. For complex media, I would highlight table 6.4. Table 6.4. And, you know, table 6.4 is short enough that I can read the list here, right? Table 6.4 is peptone, which is partially digested protein from typically a source like yeast or something like that. There's beef extract, sodium chloride, and agar, which is a seaweed extract, okay? And all that's dissolved in water. And so my point with the complex media is notice that it says beef extract. It says beef extract. So here's a question coming in the chat window. Hold on. Here we go. If each of you went to your favorite grocery store and purchased 1 pound of beef, and we sent all of those 1 pound samples of beef to a lab for chemical analysis, would the chemicals in each 1 pound sample of beef be identical? Right, you can read it faster than I can state it to you, but clearly the answer is no, right? If you got beef at Giant Supermarket and somebody else in class got beef from Wise Supermarket and somebody else in class got beef from Stauffer's of Kissel Hill or wherever you go shopping, the point of it is that those grocery stores obtain beef from a different supplier, from different animals, grown on different farms, in different geographies, under different weathering patterns, where those animals were provided different food sources. Some of them might have been treated with antibiotics, others not treated with antibiotics. The point of it is the animals are different, so the beef that comes from them is different. And so therefore the chemical composition of the beef is going to be different. So when you use complex media, you do not know the exact identity and concentration of every chemical present in the media. Okay. Because this media relies on extracts, you do not know the identity and concentration of every chemical present in the media. Okay, you do not know it. Well, so that has some implications. Because you're using extracts and you don't know everything that's in it, this makes this type of media, this type of food source, less expensive to manufacture. This is relatively cheap media. Second point, because you don't know everything that's in the media, this is not a terribly good type of media to use for scientific reproducibility. This is not the best media to use for science experiments. Okay, so I have a question for you. Think about the state of our world today. Do you think most scientific experiments use chemically defined media or complex media? Okay. All right, you get my point, gang. We live in a world that's got lots of trouble, and science as an institution, as a process of reasoning and logic, as a topic, a process that human beings carry out. Well, you know, everybody tries to stretch their dollars as far as they can go, right? You try to hold on to that car that's broken down one more year because you just don't have the money to get another car, right? Or you buy some groceries at the supermarket that are not brand name companies to try to get your money to go further. Same thing happens in the scientific lab where scientists struggle to fund their experiments. A lot of times scientists will choose the complex media, not because it's the best thing to use, but because they can make their research dollars go far when research dollars are very limiting and just about all the time. You appreciate that most of the biomedical research funded in the United States of America comes from grant proposals that are funded by the United States government coming through the National Institutes of Health. So let me ask you this in the chat window. In any given year, what percentage of research grants to the National Institutes of Health actually receive money? If you had to guess, what would you say, folks? Okay, yep. All right, thanks for those that are contributing and throwing some numbers out there. Yeah, your guesses are not bad at all. The average seems to be typically somewhere between 18 and 21%. 18 to 21% of grants submitted every year will be awarded money, which basically means that every 4 out of 5 grants that people put their heart and soul into will never get a penny, will never get a dollar. And you know, you've heard about starving artists. Well, you can hear about starving scientists now. Okay, So, I mean, it's tough all around and the money is very limited. So let me just share a quick story with you. And before I share it with you, I want to be sure you can hear me okay. Are you still hearing me okay, or am I choppy? Okay. So thank you for the feedback. When I had finished my doctoral work, I went into a period of training for scientists known as a postdoctoral training program. And during this period of time, I worked in a lab outside Washington, D.C., and I was working on a fungal parasite that kills trees. I don't know if any of you have heard, and most people honestly don't care, but have you ever heard of, I'll put it in the chat window. Here it comes. Have you heard of chestnut blight? Okay. All right. So I suspect as much. Okay. Folks, what most folks don't know is around the turn of the 1900s, the American economy, the United States economy, was largely driven by wood, and most of that wood came from chestnut trees. Chestnut tree wood was used to build railroad lines and a whole variety of other things, provide lumber for buildings and things like this. So long story short, chestnut tree wood was super mega important in the early 1900s of the United States of America. Unfortunately, around 1910 or somewhere in that vicinity, a fungus came across a ship from Europe, and in supplies from Europe, and chestnut blight was established. Chestnut blight is a disease caused by a fungal parasite that infects the wood and basically eats it and rots it out. And this chestnut blight devastated the wood industry in the early 1900s, and it caused a lot of people to go bankrupt because of it. So at any rate, chestnut blight was a big deal in the early 1900s and basically did a lot of damage to the American economy. So I was involved with research on this fungal parasite. The fungal parasite's called Cryphonectria parasitica. And so myself, working with some of my colleagues, we had some interesting results that we had got in the research lab. We took these results to my supervisor, and he looked at them and he said, yeah, this seems kind of interesting. We should probably start writing up a paper, because that's what scientists do, right? We write research papers that mostly nobody ever reads, okay? So my supervisor gave us permission to start writing a paper, but then my supervisor said those words that really every good supervisor should do. He said, well, okay, go back and reproduce the results. Make sure you can reproduce the results. Well, at the time, I was using a type of media called potato dextrose agar. This is a media that obviously is made from potatoes, and you can buy it from companies that supply researchers food sources and things like this. Long story short, potato dextrose agar is a complex media. It is not a chemically defined media. So you might ask, well, why didn't you use a chemically defined media? And the answer is because I was told not to, right? My supervisor said, use this, so that's what you use. So I used the complex media. So we went back and tried to reproduce this result we had, and lo and behold, we could not reproduce it. And it was immensely frustrating. We spent about a month troubleshooting, trying to figure out what was wrong that we could not reproduce our results. We figured, well, maybe we have a water contamination problem, because that does creep up quite a lot in the research lab. We thought, other reagents in our experiments were bad in some way. We did all this troubleshooting, couldn't come up with it, and finally it dawned on us that the bottle of food that we had used for the original experiment, we had used all that food up to do the original experiment. And when we went to reproduce, we had to open up a new jug, a new bottle of the same food. So we opened up the second jug to repeat the experiment, but we couldn't get the same results. So I contacted the company that provided this food, and I told them about the problem we were having. And I said, hey, can you find me a bottle of the original manufacturing lot that we used for the first experiment? And they grumbled a little bit and said, well, okay, we'll try to. And lo and behold, about 2 weeks later, I get a jug in the mail that had the same manufacturing lot as my original food source. And when I used that newly received jug of food, the original results returned. It came back. So there was something different between manufacturing lots that was being exposed in our research and in the results we got from our experiments. Use one kind of manufacturing lot, get interesting results. Use a different manufacturing lot, don't get those same results at all. When I contacted the company about that, they didn't have much to say other than this. They told us, well, truth be told, in between the original manufacturing lot and the second jar that you opened, we had changed our manufacturing process. And, we didn't tell anybody that, but we did that. We changed our manufacturing process and continued to label the product the same way. Okay, so this kind of crap goes on all the time in the research and scientific infrastructure communities, and it is a gigantic problem for reproducibility. Okay, so most people don't care, but this is one of the reasons why the public loses faith in scientists when you see that every second result conflicts with a previous result, right? It seems like things are always flip-flopping back and forth. What's the advice this year? Should I get a prostate check or shouldn't I? Is that good or isn't it good? Okay, I mean, the medical advice changes constantly because we have constant issues with reproducibility. Okay, so Yasmin, your question, is that legal for them to do that and not tell anyone? Yasmin, I don't know if it's legal or not, but it happens all the time. It happens all the time. And a small lab like I was in, we're not going to have the money to mount a lawsuit of any kind. I mean, it's just not worth the effort to do that. But again, it does deteriorate the reputation of science as a process that we're supposed to be able to trust. Okay, so that's my long-winded story just to show you that these issues of reproducibility are real, folks. They are definitely real. Okay, thoughts, questions? Okay, let's continue here. Number 3. So chemically defined media, best for reproducibility, often too expensive to be used, however. Complex media, cheaper, not so good on reproducibility. And we do have a reproducibility crisis in science around the world. Next, reducing media. Okay, reducing media. Thanks, Kit. You take care of yourself. Okay, reducing media is media in which the chemicals present in the media actually reduce oxygen. And I'm talking about oxidation and reduction here that we dabbled with a little bit in chapter 5. This media contains molecules that reduce oxygen. So let's talk about that. Okay, when you reduce something, if you remember our conversation from chapter 5, when you reduce a molecule, what you do is you add electrons to it, which enables the molecule to build a bond with something else. So the less sophisticated way of saying this is that this media contains molecules that basically turn oxygen into water by allowing the water to join, excuse me, by allowing the oxygen to join with hydrogen atoms. This media turns oxygen into water, okay? This is the reduction part of the conversation. Okay, and so, reducing media is under the text heading in your textbook that says anaerobic growth media and methods. Okay, and you see figure 6.6 there as an example of a chamber that would be used to grow an anaerobic microorganism. Okay, and so really that's the next talking point we need to put into your notes, is that reducing media will be used to grow anaerobic microorganisms. Anaerobes are microbes that don't like oxygen. In fact, oxygen is often lethal to them. I know that's hard for you and I to appreciate because we swim through an atmosphere of oxygen and we need it to thrive. But there are forms of microbes on the planet where oxygen is actually toxic to them and they won't live in the presence of oxygen. So reducing media is used to grow anaerobic microbes. And of course, the next talking point might be, well, that's kind of interesting, Dr. Allen, but really who cares? Because, we live off oxygen. So surely the topic of anaerobic microbes has no relevance for human medicine. And that's not true, folks. It's definitely not true. So anaerobic microbes can and are pathogens in the human body. And you might say to yourself, well, how can that be? Because once again, we're inhaling all this oxygen. Folks, you have to appreciate that there are areas inside your body that are so far away from blood vessels that basically it creates a microenvironment where the amount of oxygen is very, very, very low in that particular pocket of anatomy within your body. And it's in these pockets of anatomy within your body that have very low oxygen levels that anaerobic pathogens can grow and thrive and cause us medical problems. Let me give you some examples. Okay, there is an anaerobic bacterium by the name of Bacteroides fragilis. I'm going to type this out. Bacteroides fragilis. This is an anaerobic pathogen that causes, well, it's associated. I won't say it causes, but I'm going to say it is associated with about 90% of abdominal infections. Okay, so there are areas in the abdomen, once again, that are so far removed from the blood supply that these kind of pathogens can grow and cause problems in the abdomen. Here's another one for you. There's another microbe by the name of Prevotella intermedia. This microorganism is commonly found in periodontal disease, basically gum disease, right? If you've had gum disease or know anybody that has gum disease, if you have consistently bleeding gums, that's a warning sign of gum disease, and you really need to get to a dentist to take care of that. Okay, so commonly associated with gum disease. And you might say, well, that's ridiculous, because every time I open my mouth, I'm exposing my mouth to the air and the oxygen in the air. That's insane. How can that possibly be? But remember, folks, for those who've had A&P, remember your teeth, they fit down into your mandible and up into your maxillobones, and they're surrounded by the gum. So underneath the gum line, there is a micro-oxygen environment. There is a micro-oxygen environment, and that allows microbes like Prevotella to grow and thrive and contribute to gum disease. Let me give you another one here. There is a bacterium that has a very ominous name, at least I think it's ominous. It's called Fusobacterium necrophorum. What a mouthful, right? Fusobacterium necrophorum. And this bacterium is associated with chronic cases of sore throat. It has also been implicated in meningitis. It's also been implicated in urogenital and GI tract infections. And so let's take perhaps one of the most troubling parts of this, the sore throat. Again, you might say to yourself, well, that's insane because every time I open my mouth, I inhale air into the back of my throat. There's oxygen there. How in the world could that be an anaerobic environment? You have to remember, folks, in the back of your throat, you're going to have a lot of mucus. That mucus can accumulate in such a way that it becomes thick enough that within the mucus in your throat, there can be a micro oxygen environment that allows the bacterium to grow. I mean, remember, these are super mega tiny things. So if they can hide in a pocket of mucus, they can escape the oxygen in the atmosphere you're inhaling. They can set up shop, start growing and reproducing and making you sick. Okay. So even though we rely on oxygen, there's plenty of pockets of space in our anatomy where oxygen is very, very low, and certain pathogens sometimes can cause problems because of that. Okay, any thoughts or questions? Okay, let's continue here. All right. We need to talk a little bit about some other medias here. Let's see, number 4. I really, really wish that we could have been in lab this semester. Would have simplified a lot of things for us. Basically, on pages 162 and 163, somewhere around figures 6.9 and figure 6.10, there's some talking points that I need to get into your notes. So there are forms of media called selective media. Okay, selective media is media that has nutrients in it which promote the growth of one specific type of microbe while suppressing the growth of different kinds of microbes. Okay, so this food contains nutrients to support the growth of one specific type of microbe while interfering with the growth of a second type of microbe. Okay. All right. And so, there's many kinds of medias that you might encounter in a microbiology lab that would satisfy this characterization of being a selective media. One of those medias is something called Sabouraud media. Sabouraud. This is a media whose chemical composition favors the growth of fungi because it has an acidic pH, but generally speaking, suppresses the growth of most standard types of bacteria that we would be concerned with. So supports the growth of fungi, but suppresses the growth of bacteria, right? So obviously, if you are studying a fungus of interest to you for whatever reason, then you're going to want to use a media like Sauerbrod media because you don't want your plate getting contaminated with bacteria. Okay, so that's an example of selective media. A fifth type of media is known as a differential media. Differential media is shown to you in figure 610 there. Okay, there you have an example of mannitol salt agar. Okay. Let me mention differential media is a food source that contains nutrients that, generally speaking, promotes the growth of a variety of different things. It does not seek to suppress anything, but it does provide nutrients that allow you to distinguish different types of microbes. Okay, so this food contains nutrients that supports the growth of multiple microbes in a way that allows you to tell different microbes apart. Okay, so the example I'll give you here is from your textbook. This is an example of mannitol salt agar. Okay, so question in the chat window, folks, for those that are able to look at their textbook. Here's a question coming in the chat window. What color is the agar around Staphylococcus aureus? In figure 610, what color is the agar? Okay, thanks, Yasmin. I appreciate that. Thanks, Chelsea, everybody, Erica, Samantha, thank you. And you know what? I can see why you might say light brown. In the real world, when you kind of look at it and hold it in your hand and have natural colored light in the room, it is more of a yellowish color. But you answered correctly because I just wanted to be sure that you were looking at the right thing. So you notice the agar around Staphylococcus aureus is a brownish yellow, okay? And you notice the same agar around Staphylococcus epidermidis is a reddish pink. So you notice that both different types of bacteria grew on that plate, but one of them produced a yellow color and the other didn't. And the reason for that is because the metabolism of Staphylococcus aureus produces a molecule which in the environment of that bacterium lowers the pH of the media, and there's a pH indicating molecule in that petri dish so that when the pH changes around Staphylococcus aureus, it turns yellow, okay? So differential media allows you to basically grow everything, but still tell things apart from each other, okay? This is one of the medias that if we were together in lab, I would have had you swab the surface of your own body and swab across a petri dish, and I would have grown up in the incubator what microbes are growing on the surface of your skin. It's a fun activity to do, typically grosses people out to see what's on their skin. But folks, it doesn't matter whether you take a shower the day before or not, you got bacteria on your skin. You do, and I do too. That's just how life is on the planet. Okay. Last thing is enrichment media. Okay. Enrichment media is basically, well, let me ask you this. In the chat window, have you ever eaten at a smorgasbord? Have you ever gone someplace where, oh my God, the food and drink is just flowing everywhere, right? There's more there than you could possibly ever consume. Okay, so folks, enrichment media is a smorgasbord for microbes. That's what it is. It is a media that is very rich in a broad spectrum of nutrients. And the reason you would use this kind of media is when you're trying to grow unidentified microbes in the environment. Because if it's unidentified, you don't know what it is, so you don't know what it needs to live. So you basically try to provide it every possible food you could imagine. Okay, that's the idea here. So this is used to grow unidentified bacteria, unidentified microbes in which you don't know or understand the growth requirements for the microbe. Okay, this is a smorgasbord. All right. Okay, so if we wanted to grow something around your home and we didn't know what it was we were cultivating to begin with, we would start with an enrichment media because we're trying to grow it and we don't know what it needs, so we try to give it every kind of tasty food it might like so it has a chance of growing. Okay, gang. So that's a pretty hollow description of media, because you really start to understand media more when you're actually working in the lab. But at least we can talk about some things, and I could point you to a picture or two here in chapter 6. Gang, do you have any questions about media? Media is the food we use to grow microbes and tell one kind of microbe from another kind of microbe. Okay, let's continue here. Next is bacterial growth. Okay, so we've been hitting the topic of bacterial growth pretty hard already by doing our math problems, and all of you honestly are doing just fine with that. Even if you got the occasional wrong answer, I don't feel like we have a serious issue in class about this. So you're doing well. We need to talk about bacterial growth and reproduction, and basically that brings us to the binary fission topic of figure 6.12. Okay, so growth and reproduction of bacterial cells. Okay. All right. So you can appreciate that eukaryotic cells, like the cells that make up your body and my body, right? Our forms of cell reproduction are known as mitosis and meiosis. But bacteria don't do that, right? So bacteria do not use mitosis and meiosis like animal or eukaryotic cells do. Okay, they just don't do it. They don't have the genes for it to make the enzymes to allow those processes to occur. What they do is something known as binary fission. Okay, so binary fission, and you are aware of binary fission because we've been throwing that phrase around now all semester long without actually talking about it. Here we're going to step you through the steps of binary fission. I'm going to show you a quick little video, and it's not a terrible topic to understand. Okay, so I'm watching our time here. We're going to have to stop in just a moment, but let's take a look at figure 6.12. Figure 6.12. Okay. So when a bacterial cell is in a favorable environment for reproducing, and by favorable I mean it has access to food like decent nutrients. It is not exposed to overly harsh environmental things like ultraviolet radiation. It's not fighting a microbial war with a neighboring fungal cell that's releasing some kind of antibiotic on it. So when the bacterium is in a safe, comfortable, healthy environment, then yes, it's going to reproduce, and it's going to use the binary fission process to make that happen. Okay, so the first part of figure 6.12 is that the cell is going to elongate and DNA is going to be replicated, right? So think about it. I know I'm out of time here. If the cell is elongating, then that means the plasma membrane has to get longer, and that means the cell wall has to get longer, right? So that means you need the cell to make more phospholipids. You also need the cell to manufacture more peptidoglycan. So for the cell to elongate, there is a tremendous manufacturing capacity. There's a tremendous manufacturing and synthesis activity happening inside the cell. Okay, folks, I know I'm over here, so I'm going to pause. Okay, I'm going to pause. And okay, so I'm going to pause the recording. All right, I'll turn my video off and we'll continue. Okay. All right, gang. Thanks for the feedback. It does help me. I appreciate it. Okay, so we're talking about the reproduction of bacterial cells, more generally prokaryotic cells, but let's be frank, we know most about bacteria. We know more about bacteria than archaea. So for figure 612, we're just walking through it. Not going to take a lot of time on it because there's not too much to say here. The first part of figure 612, the cell elongates, DNA is replicated. That's going to see a time of tremendous biochemical synthesis activity, a lot of anabolic chemical reactions, right? New phospholipids getting made to expand the surface of the plasma membrane, new peptidoglycan being synthesized to build out the cell wall. Okay. DNA is going to be replicated using the process of DNA synthesis with DNA polymerase that we talked about in chapter 8 when we talked about some basic genetics ideas. Okay. The next part is that the plasma membrane begins to pinch a little bit inward. Okay, you can see that graphically in figure 612 there. The plasma membrane begins to constrict and new cell wall is made as you begin to build a wall sort of in the middle of the cell, because we're going to separate the cell roughly in the middle. And when we've completed the separation, both ends of the cell will now be physically detached from each other, and we will have two genetically identical cells. So the cell wall and plasma membrane invaginate, which is to say they pinch in at the center of the elongated cell. The cell wall completely forms a physical barrier separating the cytoplasm of basically the left side of the cell to the right side of the cell. Okay. And then, you know, the cell separates. I mean, it's kind of like And an overly simplified analogy, it's kind of like having one of those cheese logs that you might get at the supermarket, or you might get for a holiday party, a cheese log, and cutting it down the middle with a sharp knife, and you separate it, and you've got the left and right side, okay? All right, thank you, Odalis, for telling me that. Okay, all right, so the cell finally separates. Now, of course, there are things happening at the molecular level here, but much of that's really kind of beyond what we would talk about in a beginning undergraduate course. So what I'm going to do next is show you a little video that just gives you some way to think about this visually. You know, it's a little bit more exciting than hearing your old teacher talk about this topic. So here's a YouTube support video on binary fission. Let me bring this up and get the link in your notes for you. Okay, so the link is in your notes, and let me move forward with showing this video. It's just a quick 2-minute clip here, okay? Here we go, folks.
Prokaryotes are microscopic single-celled organisms, including bacteria and archaea. Prokaryotes are everywhere. If you looked, you'd find bacteria on every object around you. One reason bacteria are so ubiquitous is that they multiply very rapidly. How do they multiply so quickly? Let's look more closely at prokaryotic cell division. Prokaryotic cell division is a process by which a cell divides into 2 or more cells. The process of prokaryotic cell division is called binary fission, which is a simpler process than found in eukaryotes. Recall that prokaryotes differ from eukaryotes due to the lack of a nucleus or cell organelles. Typically, their chromosomes consist of a single loop or circle of DNA. The circular DNA molecule replicates. It then attaches each copy to a different part of the cell membrane. The cell grows to twice its size, and the membrane pinches in the middle. The cell can now undergo binary fission. Binary fission is the prokaryote form of asexual reproduction. After the cytoplasm divides, each daughter cell synthesizes a new cell wall, creating 2 identical cells. Binary fission results in 2 cells that each contain 1 copy of the original circular DNA. Prokaryotes are able to multiply rapidly because of their small size. Their DNA takes a short time to replicate, and with a high surface area to volume ratio, they can absorb nutrients quickly. Also, the relative simplicity of the cell's internal organization allows rapid duplication. A bacterium can divide every 20 minutes in optimum conditions. Unchecked, bacterial population growth is exponential, allowing rapid colonization of a favorable environment. Scientists can apply knowledge of prokaryotic cell division in disease prevention and care. Through the use of antibiotics, doctors and scientists can slow or stop the growth of disease-causing bacteria. Some beneficial prokaryotes aid in digestion. Other prokaryotes play a role in the environment and are used for cleanup of toxic waste and bioremediation programs. It's no exaggeration to say that prokaryotes are an essential part of life on Earth.
So I've got my essay written.
Okay, folks. All right, so that was just a little quick video to give you some visualization on this process. Again, it's not terribly the most exciting thing, but this is the method that bacteria use to copy themselves. Okay, bacteria are relatively simple cells. What is their life? Their life is living, and when they are capable, because the environment is supportive, they reproduce. That's what they do. Okay. All right, so do you have any questions at this time about binary fission? Okay, then we're doing pretty good here. the last talking point that I want to talk to you about in this chapter, and then we'll call chapter 6 complete, is I want to go over the bacterial growth curve. In your textbook, that's the foundation figure 615, foundation figure 615. And so let's talk about that a little bit. All right. Okay, let's see. I know I'm going to want to do some drawing, so I'm actually going to go down onto a new page here. Okay, so we talked a little bit last time, and many of you were very good at giving me some ideas about why it is important to understand something about the growth characteristics of microorganisms, right? And we talked about forensic science, we talked about the production of biological products, public health safety, food poisoning, all that kind of good stuff. And so one of the ways that scientists develop information about bacteria and other microorganisms is to develop this growth profile that's called the growth curve, okay? And that's what you see in foundation figure 615. Okay, so I want to draw that out and I want to talk to you about it a little bit. Okay. All right, so this is something that you don't really get to do this in an undergraduate lab, because most undergraduate microbiology labs are about 2 hours in length, and if they're any longer, students start to complain. But let me ask you this in the chat window. Let me ask you this in the chat window. How many hours are shown on the right side of the x-axis of this growth curve on the foundation figure 615? How many hours are on the far right of the x-axis? Right, okay, great. Thank you, folks. 10 hours, right? So it's kind of one of those things, and you know, this is more effective if we can look at each other in the face when I ask this. How many of you want to be in lab for 10 hours? I know basically nobody wants to be in the lab for 10 hours, right? But if you're going to work with microbes and living things, a lot of times when you do the work, we humans are on the time frame and the time scale of the microbes we want to manipulate. So if you want to do the science, you got to spend the time. That's just how it is. Okay. So, okay, let's take a look at this growth curve and point out important features of it. All right. Okay, so let's see, I'll use purple here, I think. So sorry, I got a wavy hand here. But you know, on the x-axis, we're plotting time in units of hours. And on the y-axis, we have the log of the number of bacteria. And let's see here. So we got about 10 hours here on the far right, 0 on the far left, and about 5 right here. Okay, so the growth curve looks something like this. You know, something like this, okay? And so let's identify the different stages of the growth curve here and talk about them, okay? So your first stage is the lag phase. This is going to be over here, okay? So stages of the growth curve. And let's be clear that we understand how this data is being generated. So to generate this data, what a person would do in the lab is they would take a piece of glass, typically an Erlenmeyer flask. And in this flask, you would put some liquid media. You would put some food in there for the bacteria, okay? You might think of it as chicken soup for the bacteria. So you put this liquid media in here, right? And then what you would do is take a Petri dish that has bacteria on it. And so the bacteria is on the Petri dish, and I'll just kind of draw it this way as a lawn of bacteria. What you would do is use typically a toothpick or some other thing like a pipette tip, which you may not know what that is, but you take a toothpick and you would scrape a little bit of bacteria on the tip of your toothpick. And you would drop that toothpick with the bacteria into the food, okay? Drop the bacteria into the food. And as soon as you do that, as soon as you drop the bacteria into the food, that is the start of your experiment. So that is time equal to 0. So you click your stopwatch at that point to turn it on and it starts counting, okay? So this is, when you drop this in here, this is time equals 0. Okay, time equals 0. And so what you do, once you have inoculated this liquid media with your bacteria, the experiment has begun. And what you will do is periodically, you will take a sample of the liquid out of this flask. And I'll just write this in words. Periodically, take a sample of liquid out of the flask and then do something to count or in some way quantify the number of bacteria present. Count the number of bacteria present. And so you will do this where, you know, at each time point that you take a sample, right? Maybe you take a sample every half an hour, right? So a half an hour is 0.5 hours. So you might have a sample taken at 0.5 hours, at 1 hour, 1.5 hours, 2 hours. You get the idea. So your x-axis is time, right? This is your x value of time. And the count that you get, the count that you get is going to be the number of bacteria at that moment of time. So maybe, and I'm just throwing numbers out here, maybe at 0.5 you have 500,000 cells. Maybe at 1 hour you have 1.2 million cells. Maybe at 1.5 hour you have 2.3 million cells. You get the idea that the cell count is the y coordinate. Okay. So let me just ask in the chat window, folks. Is this making sense what I'm describing, or have I lost you? Okay, anything you'd like to ask about the experiment? Okay, all right. So once again, you take a flask, you put some liquid food in, you take some bacteria you've grown on a petri dish, you scrape some of it off with a toothpick, you drop it into the food, you start your stopwatch, and then every so often, maybe every 30 minutes or whatever time interval you have chosen, you take a sample of the liquid out and you count the bacteria. And you make these times and the corresponding cell counts your X and Y coordinates that you're plotting on the graph, okay? And when you do that over a period of 10 hours, you typically get a curve that looks like this. So what do the stages of this curve actually mean? Okay, so I want to point out a couple things here, and you're probably going to want to look at your textbook because I'm not the best artist. But, things to think about with the lag phase. Okay, with the lag phase, and I didn't do a good job drawing, so definitely look at your book there, the Foundation Figure 615. You notice in lag phase that the slope of the curve, in the lag phase, the slope is basically 0, right? That means you have a horizontal line. Okay. So what that means is that there is no increase in the cell population during the period that we call lag phase. The population of cells is not reproducing. And so you might ask yourself, well, if they're not reproducing, does that mean they're dead? No, it doesn't mean they're dead. They're not reproducing and they're not dead. So what the heck are the cells doing? Well, here's what they're doing. The cells are making preparations to reproduce. The cells are manufacturing molecules that whenever they do reproduce, those new molecules can be given to the new cell. So the new cell has what it needs to actually live as an independent entity, as an independent cell. So you should think of the lag phase as a metabolically explosive period of time. The metabolism is going gangbusters, but we're not actually reproducing. We're not performing binary fission at this moment of the bacterial growth curve. So cells are preparing to reproduce. So I have a question in the chat window for you. Here we go in the chat window. 1 second. And here we go. What types of materials does a good parent send their child off to school with? Okay, we don't want to get political here, but what would a reasonable parent send their child to school with, right? Okay, so lunch, obviously, at least if not lunch, then lunch money, right? Hopefully completed homework, hopefully decent clothing, a backpack. Might it be reasonable to say pens and pencils, notebook paper, maybe a calculator, right? Things that the child will need to be successful in school. And so that's my point here. In lag phase, the cells that are currently living in the moment of lag phase they're not literally purchasing pencils and pens, are they? But they're manufacturing their equivalent of pencils and pens. They're manufacturing ribosomes. they're manufacturing enzymes, they're manufacturing nucleotides, they're manufacturing amino acids, they're manufacturing all the building blocks that a new cell is going to need. So that's the point. Lag phase is a very busy preparatory phase. The cell population is not increasing, but the cells are extremely busy making sure their future children, when they go off to school, are going to have the notebook paper, pencils, pens, rulers, everything that a child would need at school, everything that a new cell would need. This is where you're going to make it. And that's why cells are not going to be capable of reproducing themselves if they're in an environment that is hostile. If they're in an environment where the nutrients from the environment do not support production of proteins and enzymes, then there's no way you can begin reproduction, because the new cell would not survive because it would not get what it needs to live. So the lag phase kind of looks like nothing's happening, right? It kind of looks like nothing's happening, but that's a little bit misleading because there's a tremendous amount of metabolic activity. All right, let's continue here. Tremendous metabolic activity. Okay, any questions about lag phase? All right, then looking again at your textbook, because it's a better drawing than what I can give to you. The next phase you can see is log phase. Log phase is also known as exponential phase. I'll put it over here, log. Okay, for those of you that are somewhat, mathematically inclined, you appreciate that logarithms and exponents are related to each other mathematically. You don't have to appreciate that to do well in this course. But what you will notice here, obviously, is that the horizontal line of the lag phase is not horizontal anymore, is it? It really cranks up, and it actually has a vertical trajectory, and it actually has a very steep positive slope. Okay, so with log phase, so we have lag phase here. I should have wrote phase, lag phase. Next, we have log phase. Try not to get confused between lag and log. Lag phase, which is also called exponential phase. I will say I'm quite pleased with the coverage that your textbook gives you on some of the properties of this growth curve. You don't have to be a math guru here, but I would encourage you to read figure 613, figure 614, and try to determine what it's telling you, okay? It will help clarify some things. But at any rate, with log phase growth, you have a steep positive slope to the curve. And that positive slope tells you that there is a rapid increase in the size of the cell population. And so the only way that can happen is if you have large amounts of binary fission actively taking place, okay? Binary fission is happening in an explosive way. I have to be careful when I talk about this topic because there are some analogies that, might not be quite correct, right? Binary fission, as the video I shared with you spoke about, binary fission is a form of asexual reproduction. It's kind of like cloning, okay? There's no sex involved. There's no exchange or recombination of DNA here during binary fission, at least not typically, okay? So binary fission is an asexual reproduction. If you think about reproduction in the human body, we have mitosis and meiosis. Well, mitosis is asexual reproduction, and meiosis is a different beast, right? That's the sexual reproduction where we do swap segments of DNA between chromosomes to introduce intentional genetic variation so that our children don't look like us, okay? But with binary fission, every cell that's made basically is a clone of the mother cell that made it, okay? So binary fission is happening in an explosive way with a steep curve. And so that means that the cells that were in lag phase, they were able to organize all the building blocks that they felt they needed to give to their future children, to send their future children off to school, okay? So the cell population is rapidly expanding during log phase growth. As we get to the top of the log phase, you know, the curve starts to level off. And honestly, I've never seen a real growth curve that quite has a flat horizontal slope at the top of the stationary phase. So that's a little bit of an idealized curve in your textbook, but I think it's useful for educational purposes to talk about this. Stage 3 here is the stationary phase. Okay, so let's talk about that a little bit. Okay, so stationary phase, notice the slope of the line is basically back to 0 again. So the slope of that section of the growth curve, that slope of that section we're calling stationary phase, is basically 0, meaning a horizontal line. Slope equals 0, horizontal line. So that tells you that there is no increase in the population size of the cells, right? There's no increase in the population. No increase in cell population. So let me ask you a question in the chat window. Here it comes. Why do you think the cells stop reproducing in stationary phase? Why do you think they stop reproducing? Okay, so Yasmin, you say they are done. Well, Yasmin, why are they done?
Because they're done being reproduced,
So are you
and they're
saying-
getting
Okay.
they're getting ready for the death phase. I
But-
guess.
Okay. So Yasmin, are you telling me that the bacteria are kind of like humans, that, you know, you've had 2 children and you're just done having a family, you don't want anymore?
I think they're balanced out.
What do you mean by balanced out?
Like, I guess they got enough of what they needed, and they just stopped reproducing. I don't know, I guess.
Okay, thanks, Yasmin. I appreciate the participation. I really do. Thank you. And so just take a look at the variety of responses in the chat window, and I like a lot of what you're saying, okay? Basically here, remember, this experiment is taking place in this enclosed glass vessel called an Erlenmeyer flask, right? So this is a flask here. And so basically this is the refrigerator of the bacteria. They are living in their refrigerator. And so what's going to happen if you lived in your refrigerator? obviously, barring the fact that you might suffocate, you would basically run out of food, right? You're going to eat all the food in the refrigerator. You're going to eat all the food in the flask. So as many of you stated, they ran out of nutrients. They ran out of resources to continue to reproduce. Okay. Cells stop reproducing because of two things, not just what you said. So they ran out of food, they run out of food. Okay, so they run out of food. I think that is something that everybody can appreciate, right? If you eat all the food in your refrigerator, you don't have any more. I mean, obviously we can go to the grocery store, but the bacteria can't. So they run out of food. And there's something else. So stay with me on this ridiculous analogy, So let's say you are literally living in your refrigerator, and let's say that there is an oxygen supply. So you're not gonna suffocate, okay? But let's say you're living in your refrigerator. So I'm going to type a question in the chat window. Just give me a sec. Here we go. If you are living in your refrigerator and are eating and drinking things, what will you have to eventually do? Okay, all right. You're going to have to go to the bathroom. And the refrigerator is your bathroom. You get my meaning? In this ridiculous analogy, the refrigerator is your refrigerator, and it's also your bathroom. So here's what I'm saying. The bacteria that are eating the food, as they eat the food, their metabolism produce metabolic waste products, similar to how we form urine with our kidneys. Now, when we form urine with our kidneys, we walk to the bathroom and we relieve ourselves in the toilet. But the bacteria are trapped in this glass vessel. So they're eating their food and drinking their liquid, the water of the food, right? And eventually the metabolism, the chemistry that's happening inside there to metabolize the food and generate energy for the cells to swim around, well, you're going to generate metabolic waste products. And so what I'm telling you is that the equivalent of the microbial pee and poop gets thrown out by the cell's metabolism into the surrounding liquid food of the flask, in the liquid of the flask in the refrigerator, okay? And so basically the cells, as they live and reproduce in this environment, they reach a point where they not only run out of food, but they start poisoning their own environment from their metabolic waste products, okay? Okay, so they run out of food and they simultaneously poison their own environment with their metabolic junk, okay? And so there's one important idea here that, I would hope you can answer, but we'll see how you do here. So I'm going to put this in the chat window. Okay. What metabolic gas is made during the transition reaction and Krebs cycle that we exhale from our lungs? What's the name of that gas? Okay, thanks, Elena. Chelsea, Rachel, thank you. Everybody, it's carbon dioxide. Good. So bacteria are going to produce CO2 as well, right? Because bacteria, as you know from chapter 5, can do the transition reaction and the Krebs cycle as well. So some of the poison in the liquid media is carbon dioxide. Okay, so I'm going to put that into your notes. This includes this includes CO2 gas. Okay, now another question in the chat window. Here we go. When CO2 combines with water, what is produced? Very close, Chelsea, not quite. You're very, very close. Okay. All right, Samantha. Right. Let's take Chelsea's and Samantha's together. So H2CO3, which is carbonic acid. That's right. So as the cells in the flask are metabolizing the food sources in the liquid media, they produce CO2 and expel it from themselves into the liquid. So the CO2 gas combines with the water in the flask of the liquid media, and you form H2CO3, which is carbonic acid. Okay, so here's another question for you. If the amount of acid in the flask increases, as the amount of acid in the flask increases as cells reproduce, what happens to the pH of the liquid media? Okay, so it goes down, doesn't it? It drops, the pH value drops. So the media becomes acidic. The media becomes acidic. So the environment, the liquid environment, acidifies. And you know what I keep harping on all the time, because I harp on it all the time. Here it comes. Changes in pH change the shape of molecules. So the very molecules of the bacterial cells in this newly acidified environment change shape, they change function, and they can't do their job anymore. The cells cannot support binary fission anymore. Okay. Question in the chat window. Is this making some odd sense to you? Okay. All right, folks. So I wanted to point out to you that it's really not just a case of running out of food. It's not just that the cells are running out of food. It's also that they're literally poisoning their own environment. Okay. All right. So we'll finish this up then. As the cells transition from stationary phase, they get into that final nasty phase called death. And in death phase, which I guess I'm going to run out of notes here. In death phase, the size of the cell population goes down. notice that we do have a vertical drop in the line segment of the growth curve. And we notice that this is a negative slope, right? This is a negative slope here. Notice the negative slope. So that means there is a decrease in the cell population. Okay. And so in death phase, basically the cell population declines because the cells that were alive, some of them literally start to die. And by die, I mean the cell literally breaks open and kind of disintegrates. It's kind of like, and I don't mean literally, but it's kind of like popping a balloon. One moment you have a balloon, it's nice, full and plump with air. And then the next moment, somebody puts a pin in the balloon and it pops, and all of a sudden you don't have a balloon anymore, okay? So what I'm saying is some of the cells that were still physically alive in the stationary phase, they literally start to physically disintegrate and fall apart. Cells begin to fragment. So there is one more nuanced perspective that I should mention here. And I'm going to come back up here to stationary phase. And I need to create a qualifying statement, There's some more nuance here. Okay, so I said here that cells stop reproducing. That's not 100% true, folks, okay? So let me give you the fine print of the advertisement. The reality of it is that you have a horizontal line in stationary phase. And the reason you have a horizontal line is different between stationary phase and lag phase. You notice in lag phase, we have a horizontal line. Okay, so here in lag phase, we have a roughly horizontal line. And here in stationary phase, we have a roughly horizontal line. In lag phase, we have a horizontal line because the cells truly are not reproducing. In stationary phase, you have a horizontal line because you have some cells reproducing, but the population size is being balanced at that moment by an equal number of cells dying. So the horizontal line is not because there's no cell reproduction, it's because the amount of cell death equals the amount of cell birth. So certainly everything we said here in our notes is true. Cells are going to slowly stop reproducing because they are running out of food and because they are poisoning their environment. But suffice it to say, at an early part of the stationary phase window, let's say right here, okay, at an early part of the stationary phase window, you are still going to get some cell reproduction, but it's going to be counterbalanced by cell death. And at some point, that balancing tip is going to fall over and you're going to start coming down the curve of the death segment. And at this point, there basically is no cell reproduction happening, and it's all the cells are dying at this point. Okay, so there's this inflection where you go over the cliff, the stationary cliff, and go down into the death phase of the cell cycle, of the cell growth curve. Okay. All right. I'm getting a little long-winded here, so I probably should stop. But let me ask you, do you have any questions about this topic or this conversation? Now, I'm going to tell you folks, you're going to have questions about this topic on your exam. So would you like to ask anything at this time? Okay. Sounds like everybody's okay. So here's where we're headed, right? I mentioned that I will release the study guide for lecture exam 3 later this afternoon. You'll get it today, folks. I want to encourage you, among all the other things I know you have to do, you're busy people, is I want you to please at least do a read over of the study guide before next Tuesday's class. That way you have an opportunity to ask questions about the study guide for the exam before you actually have to take the exam. Because next Tuesday is our last point of contact before we all go and gorge ourselves with turkey on Thursday and then stumble through the weekend and come back to school the next week and take our exam, okay? So next Tuesday is the last point of contact.